Hi class and welcome to chapter 10. This chapter is on senses, sensory receptors, and all. It will focus on your cutaneous receptors in your skin and your eye and your ears. We'll first get a little introduction on sensory receptors. Sensory receptors transduce or change different forms of energy in the real world into a nerve impulse. So the light from the screen, the words, what you're hearing right now in the background, any sort of real world stimulus, sensory receptors will take that in and change it to a different form of energy so it can travel along our nerves. Different modalities of sensation arise from differences in the neural pathways throughout the brain and spinal cord and synaptic connections. So for example, if the optic nerve delivers an impulse, the brain interprets it as light, even though the impulse is the same as for hearing. The functional categories, we divide these types of sensory receptors into categories, and you should know these. Chemoreceptors sense chemicals in the environment, taste, smell, or blood. Photoreceptors sense light. Thermoreceptors will respond to cold or heat, and mechanoreceptors are stimulated by mechanical deformation of the receptor, like touch or hearing. So these are describes the receptors, the stimulus that will affect them, and then examples of each. Nociceptors are pain receptors. Stimuli can include heat, cold, pressure, or chemicals, but these are pain receptors that will depolarize or activate when tissues around them are damaged. Receptors can be classified by the type of information they deliver to the brain. Proprioceptors are found in muscles, tendons, and joints, and these tell or give the body a sense of its position in reference to what is around it and allows for fine muscle control. Cutaneous skin receptors are involved with touch, pressure, heat, cold, and pain, and the special senses are the vision, hearing, taste, smell, and equilibrium, which also occurs in the inner ear. Receptors can be classified by their origin, whether they are exteroreceptors, they respond to stimuli from outside the body, or interoreceptors respond to internal stimuli. Tonic and phasic receptors have to do with sensory adaptation. And this will explain how the receptors are categorized based on how they respond to a stimulus. A phasic receptor will respond with a burst of activity when the stimulus is first applied, but will quickly adapt to the stimulus by decreasing the response. It may deliver another burst when the stimulus is removed to provide an on and off information. This can alert us to changes in the environment, but it allows sensory adaptation. An adaptation of your senses are when, if you're focused on one thing, your senses will start to stop receiving stimulus from that in order to cease the constant stimuli. Examples of this are smell, touch, and temperature. So after a while, if you smell something, that smell might go away. It's still there, but your sensory receptors have just kind of forgotten about it. And they've done that in order to focus their attention on other new adaptations in the environment. A tonic receptor maintains a high firing rate as long as the stimulus is applied. And a big example of this is pain. So here's an example of the stimulus being applied and the stimulus withdrawn. And this is an example of a phasic receptor, which is fast adapting. This is the stimulus applied and the stimulus withdrawn in the tonic receptor. You can see how the action potentials are constant. Cutaneous sensations have to do with receptors in your skin, so pain, cold, and heat. These are specialized receptors. They're special structures around their dendrites. This is an example of the receptor, their structure, and the location, and these will all be located in the skin. So here's an example of the skin and where these receptors are located. You can see many of them here. The pachinine corpuscle is important for vibrations and deep pressure. The endings, ruthenian, are important in skin stretching. Free nerve endings will just deal with touch, pain, hot, and cold. Meissner's corpuscles will detect any changes in texture. Cold receptors, there are many more receptors that respond to cold than to hot. They're located close to the epidermis. Some of this we're actually going in lab right now, if you're with me in lab. Warm receptors are located deeper in the dermis. They're excited by warming and inhibited by cooling. And it's important to note that they're different from the receptors that detect painful heat. And hot receptors are the pain experienced by a hot stimulus is sensed by a special nociceptor called a capsaicin receptor, and it's activated at 43 degrees Celsius or higher. Pain receptors, these nociceptors can be myelinated or unmyelinated. A sudden sharp pain would be transmitted by myelinated neurons, and dull persistent pain would be transmitted by unmyelinated neurons. The itch sensation is stimulated by histamine release from mast cells and basophils. And it can be a chronic itch, which is stimulated by other chemicals, and that would not respond to any sort of antihistamines. And receptors can stimulate unmyelinated sensory axons to the spinal cord. So a lot of this, again, we've covered in our lab for this week, even if we've done, by this point, you might have done that lab a couple weeks ago. The receptive field is the area of the skin that when stimulated changes the firing of the neuron. And we talked about in lab how there are few receptors in the back and the legs, so the receptive fields are very large. And there are many receptors in the fingertips, so the receptive fields are smaller. A smaller receptive field just means there's greater tactile acuity or sharpness of a sensation. The 2-point threshold test can measure by seeing at what distance a person can perceive 2 separate points of touch. And the distance between the 2 points will be much greater where there are fewer receptors. So it's like your back or your forearm or your leg versus your fingers would be much easier to perceive the 2 points much closer together. And this gives the body region and the 2-point threshold. And again, this threshold describes the distance at which you're able to feel 2 points. So you would use some sort of device called a caliper, and it would have 2 sharp edges to it. When the points are extremely close together, it would feel like 1, and slowly you would move the 2 points out millimeter at a time until each of the 2 points would be on a different sensory neuron. And there's more sensory neurons in places like your fingertips. In places like the back of your neck, your back, there are fewer receptive fields. So this device would increase its width a lot farther before you could actually feel 2 separate points. You can try that on yourself, and it might make more sense if you try it on yourself. Chemoreceptors are interoreceptors that detect chemical changes within the body, and exteroreceptors detect changes from outside the body, like taste and smell. Taste is also called gustation, and receptors are called taste buds. And this is a look at the taste bud itself and how it'll have the gustatory taste cells, the supporting cells, and they're all connected to a sensory nerve fiber. The taste bud is located in bumps on the tongue called papillae, and there's different types of papillae. Fungiform are on the anterior surface, circumvallate are on the posterior surface, and foliate papillae are on the sides. The taste pathway, the taste goes through the facial and glossopharyngeal nerve. So that's the sensory neuron. It goes up through the medulla oblongata of the brainstem into the thalamus, where it's taken into the primary gustatory cortex of the insula, the somatosensory cortex of the parietal lobe, and the prefrontal cortex. The taste cells of the taste buds are specialized epithelial cells. They have microvilli that come into contact with chemicals, and each taste bud has taste cells sensitive to each category of tastes. These are the 5 different types of tastes that I'll let you read through there. And again, this little bit of this is kind of a review from lab. You don't need to know how each specific taste is transmitted to the sensory neuron. This is how taste works, a salty, sour, sweet, umami, or bitter taste. Basically, there's sodium or some sort of device that will enter, ion that enters the taste cell to cause a depolarization of it. and that will send the signal off through the neuron. The olfactory apparatus has to do with smell. Smell is also called olfaction. Olfactory receptors are located in olfactory epithelium of the nasal cavity, and these olfactory cells will eventually lead up to the olfactory nerve. So here is the olfactory bulb, the olfactory tract, or that olfactory nerve. And if you remember from anatomy, here's your cribriform plate of your ethmoid bone, and it has tiny holes through it that these olfactory sensory neurons will travel through. And the ends of them have little cilia on them, and they will detect any sort of chemicals or things you breathe in, and the cilia will absorb them and get sent up through this pathway system to the brain. Olfactory receptors are a special kind of neuron called a bipolar neuron. They have ciliated dendrites where proteins in the cilia will bind to the odors, and one type of molecule will stimulate one protein. And this is a look at those cilia that will bind to each odor. It's kind of a cool picture. The vestibular apparatus and then equilibrium. This has to do with your ear. The vestibular apparatus provides a sense of equilibrium throughout the entire body. It's located in the inner ear. It consists of the otolith organs, which is the utricle and saccule. These help the body balance in linear acceleration, and the semicircular canals help the body balance in rotational acceleration. So here are the semicircular canals in the utricle and the saccule, and where they're located in the inner ear. Here's the cochlea, which sound waves will eventually go through. And here's the vestibular nerve and the auditory nerve. They're two different branches of the vestibular cochlear nerve, one of the cranial nerves. The inner ear consists of a bony labyrinth surrounding a membranous labyrinth, and between the two is a fluid called perilymph. Within the membranous labyrinth is a fluid called endolymph, and endolymph usually has a high potassium concentration, which will produce a depolarization. So here's just a look at the semicircular canals, the semicircular ducts of the membranous labyrinth, and then the connection to the cochlear duct. And here's the cochlear duct with the cochlea, all filled with fluid. Sensory hair cells are modified epithelial cells with 20 to 50 hair-like extensions called stereocilia. And these stereocilia will bend with sounds that pass through the cochlea. And as they bend, that bending produces an action potential, and that bending will produce a different frequency of sound, whether it's stimulated or inhibited, but it's the bending of these stereocilia hair cells that the nerves connected to them will pick up as sound in different pitches. So this kind of takes you through how that occurs when stereocilia bend. Potassium channels open and potassium rushes into the cell, depolarizes it, and the cells will release a neurotransmitter that depolarizes the sensory dendrites. Bending away from the kinocilium hyperpolarizes sensory dendrites, and this becomes a code for detection of direction. The utricle and saccula then are that organ that help provide information about linear acceleration, whether it's in the horizontal or vertical axis. There are specialized epithelium called the macula, which houses more hair cells, and these stereocilia hair cells are embedded in like a gelatinous fluid in the otolithic membrane. The gel also will contain crystals of calcium carbonate. So this shows if you bend your head downward, these hair cells will bend, and that bending of these stereocilia will be detected by these nerves. So the easy way to remember, throughout your ear, there's tiny hair cells called stereocilia, and the bending of these stereocilia is what causes the nerves to pick up any sort of stimulation, whether it's in rotational movement, whether it's in equilibrium, or whether it's in sound. The semicircular canals project along 3 planes to detect rotation, Each canal contains a semicircular duct filled with endolymph, and at the base of each duct is an enlarged area called an ampulla. Hair cells are embedded in the crista ampullaris with stereocilia stuck into a gelatinous cupula. So rotation, you can see here, rotation on his axis will cause bending of these hair cells. And again, whenever these hair cells will bend, and again, they are embedded within the supporting cells, the bending of the hair cells will produce a stimulus in the nerves that they're connected to. And that stimulus will go off to the brain and tell the brain what's happening in the inner ear. So how do ears hear? We talked a little bit about equilibrium, balance, but let's talk about how we actually hear. Sound waves are characterized by frequency measured in hertz and the intensity or loudness measured in decibels. So this is a review of anatomy from the ear, and you should have a good understanding of this because each part that you learned in anatomy will directly kind of be pulled together to explain how sound works. Really simply, sound will come in through the auricle and through the external auditory meatus. Sound will cause vibrations against the tympanic membrane, and those vibrations are amplified through these three tiny ossicle bones. As these bones vibrate, they produce kind of a pounding sensation on these organs of the inner ear. And that pounding produces, remember within the inner ear there's fluid, and the vibrations will travel through the fluid, causing the fluid to move through the cochlea. And those vibrations moving through the fluid in the cochlea and through the semicircular canals and the other organs will cause those teeny tiny hair cells to bend. And that bending is picked up by branches of your vestibular and cochlear nerve. The auditory tube just helps to equalize pressure. So when you're in high altitude or in an airplane and you hear your ears pop, what you're actually hearing is your tympanic membrane, your eardrum popping back into place. And your auditory tube helps with equalizing pressure because it opens into your nasal cavity. So sound waves, I kind of just explained this, so I'll let you guys read through. You can pause the PowerPoint or the lecture video and read through these slides to get a more in-depth look at what I just talked about. The middle ear, sound waves will vibrate against the tympanic membrane and cause these ossicles to vibrate against the oval window, which is the base of the stapes bone. And then the cochlea is the hearing part. It has three chambers. The upper chamber is a portion of the bony labyrinth called the scala vestibuli. There's also a lower bony chamber called the scala tympani, and both chambers are filled with that perilymph. The cochlea also contains a portion of the membranous labyrinth called the scala media, or the cochlear duct, and that's filled with endolymph. And here's a look at the cochlea if we were to take a cross section of it. So again, the cochlea looks like a snail shell. And if we take a cross section of it, you can see all like the winding kind of canals of it, and how each canal has a top scala vestibuli, bottom scala tympani, and the cochlear duct in the middle. And within this cochlear duct, you'll see the spiral organ of Corti. The base of it will be the basilar membrane, and embedded in this area of the spiral organ will be those stereocilia cells. So sound transmissions, vibrations from the oval window of the middle ear will displace the perilymph in the scala vestibuli. Vibrations will pass through the vestibular membrane into the cochlear duct through the endolymph, and next vibrations will pass through the basilar membrane into the perilymph. Vibrations will leave the inner ear via the round window. So they entered from the oval window and they will leave via the round window. Sound waves are transmitted through the cochlear duct at locations that depend on the frequency. Low frequency sounds just travel further down the spiral of the cochlea to the apex, and higher frequency sounds are closer to the base. So if we unroll that snail shell and we just roll it completely out, we can see how the vibrations enter through the oval window, and again they're being produced from the vibrations that are coming through the ossicles. The vibrations displace the fluid in the perilymph, and the vibrations will eventually exit out of the round window. And you can see here, depending on the frequency of the sound, in red, green, or blue, lower frequencies in blue will reach all the way out to the end or the apex, and higher frequency sound will bend the stereocilia cells closer to the base. Sensory hair cells are located on the basilar membrane. They project into the endolymph of the cochlear duct, and each of these inner hair cells is innervated by 10 to 20 sensory neurons of that cranial nerve, the vestibular cochlear nerve. These hairs are stereocilia that are large microvilli arranged in bundles. They are embedded in the gelatinous tectorial membrane. And again, the spiral organ is made up of the basilar membrane at the bottom, the inner hair cells with the sensory fibers, and then the tectorial membrane that they are embedded into. This is how hearing works. Sound waves will enter the scala media. The tectorial membrane will vibrate, bending those hair cells or the stereocilia. And when those stereocilia bend, that causes the depolarization of the neurons, and that's what will take the stimulus up to the brain. The greater the amount of basilar membrane displacement and bending, the more glutamate that will be released, and that will produce a greater receptor potential. So this is a great, I'm hoping you guys had some of this in anatomy, but this is a great review of anatomy here. Here's the basilar membrane, and then the tectorial membrane is at the top. But these are the nerve fibers and how they're embedded. You have inner and outer hair cells, but they will be embedded in the tectorial membrane, and the bending of those hair cells will be picked up by the vestibular cochlear nerve. Detecting sound frequency. This goes through the place theory of pitch, and I'll let you guys read through that on your own. I won't ask you too many questions about this. I'm just going to ask for a basic general understanding of how sound works. Here's the portions of the cochlea that detect different frequencies. And this is the neural pathway. So there is a neural pathway for smell. There will be one for vision. The neural pathway will, the sound sensations waves will be coming through the vestibular cochlear nerve. They'll go up to the medulla oblongata and the pons of the brainstem. They'll go to the inferior colliculus of the midbrain, and then they'll go to this body region of the thalamus and into the auditory cortex of the temporal lobe. So here shows the neural pathway traveling through the medulla oblongata, where they will cross over, and then they'll travel up through the midbrain and then to different regions in the thalamus. I won't ask you guys too many specifics about neural pathways. Hearing impairment, conduction deafness, our sound waves are not conducted from the outer to the inner ear, and this could be due to buildup of earwax, too much fluid in the middle ear, damage to the eardrum, or overgrowth of bone in the middle ear. This will impair hearing of all sound frequencies, but it can be helped by a hearing aid. If we get into sensorineural or perceptive deafness, this is when nerve impulses cannot be conducted from the cochlea to the auditory cortex. And this can be due to damaged hair cells from loud noises, so those stereocilia have been damaged. It may only impair hearing of particular sound frequencies and not others. Cochlear implants could help with this. And presbycusis or cussis is age-related hearing impairment. My dad has that pretty bad. He does not hear very well. So eyes and vision are kind of the last part of this, so feel free again to take a break if you need to. And then this will be kind of the last section of this chapter 10 that we'll go over. Vision comes from light energy transduced in nerve impulses. So light comes in through your eye and it's transduced into nerve impulses on the retina, the nervous layer of the eye. Only a limited part of the electromagnetic spectrum can excite the photoreceptors. So that is why these are the colors that we see in visible light. This is a good review of anatomy. So I'd encourage you to pause the video here, use this slide to read through the review of anatomy, and use this to review it as well. The structures of the eye, this takes you through some more review of the eye. light will pass through the cornea and into the interior chamber of the eye where it passes through the pupil, which can change shape. And the pupil changes shape due to the iris muscle to allow more or less light in. A dilated pupil will allow more light in, and a constricted pupil will allow less light in. More general pathway of light as it hits the eye. After the pupil, it will pass through the lens, which can change its shape to focus the image. It will pass through the posterior chamber and the vitreous body, which is the gel-like substance that fills up the posterior chamber. And finally, light will hit the retina, the back of the eye where photoreceptors are found. And then that light will be absorbed by the pigmented choroid layer. Here's the pupil and iris. Again, the iris is the muscle that controls the diameter of the pupil via constriction and dilation. Parasympathetic stimulation constricts the eye, rest and digest, and sympathetic stimulation dilates the eye, your fight or flight. You should know which autonomic nervous system constricts the pupil and which autonomic nervous system dilates the pupil. The iris also has pigmented epithelium for eye color, and that's what gives us all our different eye colors. So here you can see the different layers of muscle in the iris and how each one works to dilate the eye or dilate the pupil in dim light and constrict the pupil in bright light. The lens is avascular. It will be attached to muscles called ciliary bodies and suspended from suspensory ligaments. A cataract is an area of cloudiness in the lens, which is normal transparent. It can be damaged from UV light, dehydration, or oxidation. Cataracts will interfere with vision in over half the people over 65 and are often treated by surgical removal of the lens and replacement of an artificial lens. So here's a relationship between the ciliary muscles and the lens. Here are the suspensory ligaments holding the lens and the ciliary muscle connected to those suspensory ligaments. And again, these suspensory ligaments connected to the ciliary muscle will control the shape of the lens to just allow for better focusing of the light. The aqueous humor fills the anterior and posterior chambers. It's a clear watery liquid. Inadequate drainage of this will lead to glaucoma or increased pressure. Here's the production and drainage of that aqueous humor. And again, yep, we'll talk about glaucoma right now. It's a damage to your optic nerve. Open angle glaucoma is the most common. It's inadequate drainage, so we get intraocular pressure that can arise above normal. It's painless, it lacks symptoms, except for the gradual loss of your peripheral vision, and its progression can be treated with medicines and some surgical procedures. The angle closure glaucoma is caused by the sudden rise in intraocular pressure due to a blockage of the angle by part of the iris, and this must be treated immediately with laser surgery to prevent blindness. So light refraction just means bending. When light passes from one medium to another with a different density, it bends. Curvature at the point of refraction can also bend the light, and the greatest refraction is at the air-cornea interface. The humors will also contribute to the refraction. Changing the curvature of the lens allows for fine control of focus. So your lens will be constantly changing its shape to focus the light. The image that comes into your eyes will always be flipped upside down and right to left in this process. So here is the area of vision and how this area of vision will come in and then be bent through the lens and then upside down on the back of the eye. The visual fields are part of the external world projected onto the retina, the back of the eye. The right side is always projected on the left side of the retina, and the left side is always projected onto the right side of the retina. So this just shows the field of vision and how the left field of vision coming into the right eye will be projected on the right side of that right eye and vice versa. So if you kind of just cover up one half of the screen, just look at the right eye right now, focus on everything your right eye sees. What your right eye sees in the right field of vision will be projected to the on the right field of vision will be projected to the left side of the eye in the retina, and what it sees out of the left side of the eye will be projected onto the right side of the eye. And then how that takes, you'll see kind of how it crosses at the optic chiasm, where your optic nerves cross, and you'll see how everything from the right field of vision, it's projected onto the left side of the retina and goes up to the left side of the brain. Everything from the left field of vision in yellow will be projected to the right side of both retinas and will be taken up to the right side of the brain. Accommodation is the ability of the lens to keep an object focused on the retina as distance between the eye and the object moves, and this is due to contraction of ciliary muscles. When the ciliary muscles contract, this allows the suspensory ligaments to relax and the lens to thicken and round up. This is good for extremely close vision. When the ciliary muscles relax, this pulls on the suspensory ligaments, causing the lens to be thin and flatten, and this is good for distant vision. The near point of vision is the minimum distance from the eyes that an object can be brought into focus, and presbyopia is the loss of the accommodation with age. You lose the flexibility of the lens due to the forward movement of these zonular fibers in the suspensory ligaments. So the lens cannot thicken to increase refraction for near objects. So that's why people need reading glasses. So this is a look at, again, at the lens, and again, the attachments, the zonular fibers of the sensory ligaments are what will cause the lens to change shape. Here's lens accommodation, and I do want you guys to know this. The lens accommodation, when the ciliary muscle fibers are relaxed, the sensory ligament is taut, and the lens is thin and focused for distant vision. And then when the ciliary muscle fibers are contracted, that causes the suspensory ligament to relax, and the lens will be thicker and focused for close-up vision. So I want you guys to understand how the ciliary, how all of these relate and how they change during lens accommodation for looking at things distance and for looking at things close-up. Visual acuity is just sharpness of vision that depends upon resolving power. It's measured at 20 feet with the Snellen eye chart. Myopia is nearsightedness. Distant images are brought to a point of focus in the front of the retina, and it's often due to elongated eyeball. It's corrected by a concave lens in eyeglasses. So myopia is nearsightedness. This is what most of us have if we wear glasses or contacts. We have hard seeing things further away. Hyperopia is farsightedness. Distant images are brought to a point of focus behind the retina. It's often due to a short eyeball. And astigmatism is where the asymmetry of the cornea and or lens curvatures is off. You can have a very sharp or steep lens. You get several points of focus on the retina. It can be corrected by different types of contact lenses. Hopefully they'll show you what. So here is myopia, nearsightedness, and how it can be corrected. Hyperopia is farsightedness, and astigmatism is raised do not focus in general. For astigmatism, if you're looking at kind of a clock and you look at radiating lines going out from the center, all the lines would appear a little blurry. And emmetropia is just normal vision where no correction is necessary. So again, you should know the difference between myopia is nearsightedness. That's what most of us are who wear glasses. We have trouble seeing things far away. Hyperopia is farsightedness. It's harder to see things close up. The retina then, it's a forward extension of the brain, so the neural layers will face outward toward the incoming light. Neuron axons in the retina are gathered at a point called the optic disc. This is the blind spot, and exit as the optic nerve. Blood vessels also enter and leave here. So at the blind spot, you will have no photoreceptors. So everyone has a blind spot in their eye. So here's a fun picture of the retina. Here's the optic disc where the optic nerve will attach, and you can see the blood vessels coming out of it. So there are no photoreceptors there. Here's the macula lutea, this area with the Fovea Centralis in the center of it. And that's where the concentration of cones will be greatest. So this is the area of sharpest vision. Photoreceptors, rods and cones are in the inner layer. These synapse on a middle layer of bipolar cells, which will synapse on the outer layer of ganglion cells. And I will just, this is a kind of a great picture to describe how light comes in, and the light goes all the way back to synapse on the photoreceptor cells, and then it will synapse on the bipolar cells, and eventually into the ganglion cells of the optic nerve. So the direction of light actually goes through kind of everything first, and synapses in the back of the retina first by the photoreceptor cells, and then makes its way back up to the bipolar ganglion and fibers of the optic nerve. Rods and cones consist of the outer segment full of flattened discs with photopigment molecules and an inner segment that contains the cell organelles. They're named rods and cones because a cone has a more cone-like shape and a rod looks more thicker in shape. The retinal pigment epithelium is a single layer of cells located under the rods and cones. This is how it helps vision, and I'll let you read through that. Rods will allow for black and white vision in low light. They contain the purple pigment rhodopsin, which absorbs green light the best. So rods are associated with the ability to see in low and dim light. The visual cycle of retinal, you don't have to know this. Here's a look at the different associations of rhodopsin. You don't have to know this. All you need to know, and I want you to know, is that when rods help us to see in dim light, so for example, when a person enters a dark room after being in the light, there are fewer photopigments in the rods and cones. But after about 20 minutes, more visual pigments will be produced, and the person's eyes will adapt to the dark due to the structure of the rods. And that is why if you're in a dark room, you eventually can see better after a period of time. Bipolar cells can stimulate the ganglion cells. So how this works, in dark and in light, the light will be absorbed, and then after the light will be absorbed by the photoreceptor cells, and then the photoreceptor cells will be able to stimulate the bipolar and ganglion cells. So this is the basic structure. Light comes in, is picked up by rods and cones, and stimulates the bipolar and ganglion cells. And the ganglion cells will be what are connected to your optic nerve. Cones are less sensitive to light, but they allow for color vision and greater visual acuity. There's 3 different types of cones in what different wavelengths they perceive, and that will have to do again with what colors they'll perceive then. And instead of the opsin pigment, photopigments have photopsins with retinine, and photopsins vary in each type of cone depending on the color. Color blindness is caused by the congenital lack of one or more types of cones, and is more common in men than women, as these genes are on X chromosomes. And most commonly, this involves either the red or green cones producing red-green color blindness. So many people just are colorblind to certain colors. And again, it most often affects men. If we were in class, we have little colorblind books that you could test yourself. People with this condition have only 2 functioning types of cones and have trouble distinguishing red from green, and see colors like purple and brown, which are combinations of colors. So these are the 3 different types of cones, and again, S, M, and L describe the lengths of their wavelengths. Visual acuity and sensitivity. Vision is best at one point in the retina called the Fovea Centralis, and you should know that. Each cone has a 1-to-1 relationship with a ganglion cell, meaning each cone is directly attached to a ganglion cell, which allows for greater acuity. It only works in good light. Convergence of lots of rods onto a single ganglion cell increases light sensitivity. So this is just showing how the cones in the Fovea Centralis have a direct 1-to-1 connection with the ganglia. And this is showing how light comes in, cones in the fovea, and this might be a better picture than I just showed you, but do you see how rods might converge onto one ganglia, whereas cones have a one-to-one connection with each ganglia, and that which gives them greater visual acuity in that area. Here's age-related macular degeneration. It involves degeneration of the macula lutea and its central fovea, so it'll affect 1 in 3 people by the age of 75 and is the leading cause of blindness. People with macular degeneration lose the clarity of vision provided by that fovea where your cones are, and 30% of the vision in the central region. The damage is usually related to the loss of the retinal pigment epithelium. Here's the neural pathway for vision. And again, we kind of talked about this already, but anything that comes into the line of vision is put on the opposite side of that eye, and then that side of the eye will take it to that side of the brain. The control of eye movement, there's produced by contraction of 6 extrinsic eye muscles, and this goes through the 3 types of movement, which I will not ask you questions on. The pupillary reflex, this will be important to control how much light goes to the back of the eye. It's controlled by, it will be constricted by the parasympathetic reflex, and your pupils will be dilated by the sympathetic reflex. It will be suppressed in the pineal gland. And that is all I have for this PowerPoint. I took out some slides, trying to make it a little more concise. Thanks for listening, guys.